Approximately 1 in 3 humans on Earth carries a highly successful pathogen in their body without ever realizing it. This is Toxoplasma gondii, which is an obligate intracellular protozoan parasite. For most people, a healthy immune system forces the parasite into a dormant state, suppressing any symptoms. But for the immunocompromised, the infection can cause severe organ and brain damage. It also poses a severe threat during pregnancy. If a mother acquires a primary infection, the parasite can cross the placenta, causing devastating neurological and ocular damage to the developing fetus. Emerging research suggests the stakes may be even broader. Clinical studies are finding correlations between latent toxoplasma infections and psychiatric syndromes, including schizophrenia. This highly effective, neurologically active pathogen is successfully hiding within a third of the global population. Toxoplasma has a highly specific reproductive constraint. It requires a definitive host to complete its sexual reproductive stage, and biologically, that can only happen inside the intestines of felines. Herein lies the core biological paradox. The parasite strictly requires a cat to reproduce, yet it has managed to infect 2.6 billion humans and countless other warm-blooded species. It has even infiltrated environments completely devoid of felines. Off the coast of California, endangered southern sea otters are contracting and dying from virulent, atypical terrestrial strains of Toxoplasma. These marine mammals acquire the pathogen because the parasite's reproductive spores survive the journey from land waste through terrestrial watersheds, eventually discharging directly into the ocean. To understand how this pathogen became so ubiquitous, we have to look past the cat itself and trace the massive environmental transmission matrix it leaves behind. This life cycle diagram illustrates the transmission web, starting at the top with the definitive feline host. When a cat acquires a primary infection by eating infected prey, it sheds millions of microscopic oocysts in its feces for up to 3 weeks. However, those oocysts are not immediately dangerous. They require an incubation period of 1 to 5 days in the environment to sporulate and become infectious. Because of this shedding process, medical professionals often advise pregnant women to surrender cats to shelters. But feline-acquired immunity makes this precaution largely unnecessary. Cats stop shedding oocysts after their very first exposure. Prolonged or repeated shedding is exceedingly rare. Furthermore, because of that 1 to 5 days sporulation delay, a daily routine of cleaning an indoor litter box breaks the cycle before the oocysts can even become infectious. Blaming domestic cat contact is simply an epidemiological distraction. It completely ignores the true environmental vectors that are driving this massive global infection. The real danger lies in the parasite's extreme environmental resilience. The sporulated oocysts can survive for highly extended periods in both soil and water, easily withstanding standard wastewater treatment processes. When contaminated cat waste is flushed down a toilet, resilient oocysts pass right through treatment plants and empty into the ocean, bridging the gap to marine mammals. On land, the oocysts settle into the soil and water, where they are ingested by livestock, by small rodents, and foraging birds. Once inside these intermediate hosts, the parasite shifts its strategy. It immediately begins rapid asexual replication, creating millions of clones called tachyzoites that disseminate heavily throughout the host's bloodstream. To evade the host's immune system, these tachyzoites entrench themselves, forming heavily armored, dormant tissue cysts known as bradyzoites, primarily within muscle and brain tissue. Humans contract the parasite primarily through 2 environmental vectors: ingesting resilient oocysts from unwashed produce or contaminated water, and consuming bradyzoite tissue cysts in undercooked meat. Toxoplasma gondii achieves its massive global prevalence by exploiting our agricultural systems and water supplies, bypassing the need for direct feline-to-human contact entirely. With tens of millions of people infected in the United States alone, the obvious medical question is why we lack an effective human vaccine. The only commercially available option, Toxovax, is a live attenuated formulation. It works for sheep, but it is biologically unsafe for human application. To achieve sterile immunity in humans, a vaccine must engineer a massive Th1-type cellular immune response, specifically driven by interferon gamma molecules. The immune response fails because these interferon gamma molecules are effectively repelled or ignored by the dormant bradyzoid tissue cysts. To solve this, researchers are zooming into the parasite's membrane, identifying multi-stage conserved antigens, specific surface proteins labeled SAG, GRA, ROP, and MYC. Current development relies on advanced mRNA and adjuvanted subunit platforms designed to specifically target these exact antigenic proteins. The parasite's biological ability to lock itself in a dormant bradyzoid stage is the exact mechanism that has stalled human translational medicine for decades. If the parasite's biology makes humans too difficult to vaccinate directly, the medical strategy requires a pivot. We have to change the target. Researchers are proposing a One Health deployment strategy, treating human, animal, and ecological health as a single, integrated matrix. Returning to the life cycle diagram, the first practical goal is to vaccinate livestock using mRNA platforms, directly reducing the bradyzoid tissue cyst burden in the global food supply. The second goal focuses on the definitive host. Vaccinating cats permanently interrupts the initial shedding of oocysts. Eliminating these transmission nodes creates a protective firewall around human and marine populations downstream. Defeating an obligate parasite doesn't require curing the victim. It requires engineering immunity within the biological engines that drive its spread.